So we will continue with the session on challenges of nanoparticle with host interactions. Now it's very clear that when it comes to being exposed to nanoparticles like viruses, the immune system is still the best medicine we have available. Yeah? And so it's a pleasure to invite Professor Gerrit Borchard from the School of Pharmaceutical Sciences of the Universities of Geneva and Lausanne to give his presentation.
Thank you, Professor Paro, and very excited to be here today. We'd like also to thank Beat and Patrick for having me here today. Thanks a lot. I hope that, talking about excellence in science, I hope that Dr. Häfliger is still with us now, because I work at the School of Pharmaceutical Sciences, Geneva, Lausanne, and according to a recent ranking of schools of pharmacy and departments of pharmacology, Geneva, Lausanne ranks first in Switzerland, first in the French-speaking world, 8th in Europe, and 21st in the world. So that's not half bad. I am working in biopharmaceutical sciences, as I said. What always interested me was this threshold between the science or art of making formulations, drug formulations, and the biological systems these drug formulations have to work in. So really pharmaceutics and biology there. So going to Biopharmaceutics 101, I think you recognize this. So you have a drug, you have a target, you have an effect. It's not as simple as this. Of course, you have off-targets, and thereby you have side effects. Now, going a little deeper into this, because this is, of course, not the whole story, you have what the physical-chemical or pharmacokinetic properties of the drug, ADME-T, so absorption, distribution, metabolism, elimination, and what comes out there is also the toxicity. So this is governing what we call PK/PD here in the end. Now, with some tricks of pharmaceutics, and you can change these, the ADME-T by, these ADME-T is of course governed by physical-chemical properties of the drug. However, you can change this by formulating these drugs, make it more soluble, make it more penetratable for the permeable for application and so forth. So this is basically the picture. This works very nicely. You can very nicely describe this for very simple structures like aspirin here, but also for very complicated structures that the organic synthetic chemists can come up with in their labs. Now, switching gears to nanomedicines, and they do not always look like these particle-shaped or football-shaped particles you see here. They come in all shapes and sizes. You have these liposomes, you have the polymer conjugates, you have, so I depicted here some of the glutaromoids. These are mixtures of polypeptide chains, very heterogenic. So all these can be summarized to be nanomedicines. Now, how do you describe this? Does this biopharmaceutical principle still applies to this? Now, I would say, what is the drug in these NBCDs, these non-biological complex drugs, these nanomedicines? Is this the vector plus the API, or is it the API plus the vector, which is actually the drug? Now, can we describe the physical-chemical properties of like these constructs here, of these very heterogeneous mixtures of nanomedicines? Now, and what happens if you formulate the whole stuff, you freeze-dry it, you put it into something else, into an emulsion? Do you change the properties of these nanomedicines? Sometimes the ADME-T, of course, is hard to describe. For some of these glutaromoids, for instance, there is no proper pharmacokinetic profile that you can describe to characterize the drug. Then there is the target, sometimes it's not known, and you have off-target effect and side-target effect. So a lot of question marks, and there's one thing that we need to consider as well, as these are particulate structures. There's the immune system that plays a role, which does not play a role so much, but to that extent, for small molecular weight drugs. Now, talking about non-biological complex drugs, these are synthetic, of not a biologic origin. They're large molecular nanoparticles, maybe some polymeric mixtures. And here the entire complex, so the API plus the vector, whatever it is formulated in, is the active pharmaceutical ingredient. And these properties cannot be fully characterized by the physicochemical analysis alone, and we need, and then same as for therapeutic proteins, we believe that the manufacturing process is fundamental to create the product, and this process is very difficult to control. Now here's some examples of these NBCDs. There's these liposomes. I think some people know them very well. There are these glyceridoids, which are a polypeptide mixture of these four amino acids, a very heterogeneous mixture of these peptides. And here these are the iron sucrose or iron carbohydrate drugs. So basically iron nuclei which are surrounded by a matrix of carbohydrates, mostly iron sucrose. Now what can happen if things go wrong, if you use the wrong formulation parameters, is shown on the next slide. Now this is a study that was published by Jacques Rotembourg from Paris in 2011. What it shows you is, in the phase 1 of these patients, hemodialysis patients, chronic renal failure patients, they were treated with Venofer, which is the iron sucrose originator product. And then the HB values, the hemoglobin values, were quite nice. You have some erythropoietin that you have to give, some additional iron you have to give to these patients, but they were quite stabilized over this first phase 1, this first period here. Now something happened here in between. The administration of the hospital where these patients were treated decided to change to a similar, iron sucrose similar, or follow-up, or however you want to call it, without even informing the attending physician. What happened is that the patient's HB values dropped dramatically here. So what they needed to do is increase the IV iron drug during the phase 2 to come back to the normal values here. They had to increase by 35%, and they had to increase erythropoietin by 12.6%. Of course, the switching was because they wanted to save some money, but in the end it turned out not to be cheaper to use the iron sucrose similar. So obviously, even with these small iron particles surrounded by sucrose, there is something going on. It's really complicated to make these drugs, and if you change some properties, you have in the end some different clinical outcomes. The next slide shows you also some adverse drug reactions, this time in postpartum or gynecology surgery patients for the iron sucrose original and for a similar. I'd like to point out just these injection site reactions. For the iron sucrose original, you have something like 2% of ISRs. If you switch then to a similar, this is much higher. And even if you do some formulation here, you only dilute the whole thing with 100 milliliters of saline, you get even a higher increase in injection site reactions. So in contrast to what you expect, because these are more higher concentrated, you may have some higher aggregate contents in these high concentration solutions. If you dilute it, then you would think that you get less aggregates or less ISRs, but on the contrast, you see an increase in these ISRs for the diluted concentrations there as well. So nanomedicines are not easy, are not simple concepts there. Now this was also now seen by the FDA. In 2013, they gave away a study to study the therapeutic equivalence for Nulicid and Ferlicid to iron carbohydrate drugs, one the originator, the other one the follow-up. And they included here a number of things to be looked at. This is now given away to a group in the US, but also to look in patients, not only do the in vitro test, not only do the physical chemical characterization, but they insisted on having some verification in patients as well. Now what we think, and I work here with a group, the non-biological complex drug group managed out of the top institute pharma in the Netherlands, in Leiden in Holland. We think that for giving approval to nanosimilars, so follow-up drugs, similar drugs to nanomedicines, we think there is something missing in the regulatory process. So for generics, for small molecular weight drugs, they're fully characterized. You can, have the molecular weight, whatever physicochemical properties you want to know. You do some bridging studies. Voila, you have your generic drugs. Now for the biosimilars, for therapeutic proteins here in Europe, we are quite okay. The first 2 biosimilar drugs were actually brought onto the market by 2 Swiss companies. Now we have about, I think, a dozen biosimilars in the European market. The FDA is still a little more reluctant. They're still discussing how they want to attack this. However, this is a regulatory process that is there, which recognizes that these biological drugs cannot be fully characterized. So what you need is clinical studies. If you want to do a biosimilar, you have to have post-marketing surveillance and so forth and so forth. So we now think for these similars, or these non-biological complex drugs, we need to do the same regulatory approach or something like the regulatory approach like for biosimilars. Because these drugs cannot be fully characterized only by physicochemical means alone. Now, this was also led to the installation of a working party for non-biological complexes, or NBC, at the European Directorate for the Quality of Medicines, EDQM, in Strasbourg, which is now tasked to develop a monograph for iron sucrose concentrated solution as a first template for a number of different nanomedicines, nanoparticulate drugs. The other thing is that what's happening at the EMA level here in Europe is that they published a draft of a reflection paper on the data requirements for intravenous iron-based nanocolloidal products developed with reference to an innovative medicinal product. So in a way, they're thinking the same lines that this regulatory approach should go in the same direction as for the biosimilars. This draft paper was out for consultation from September 2013 to February this year, and we're waiting for the feedback on the feedback we gave them for this draft paper. Switching gears now. Patrick, I think, mentioned vaccines. Vaccines where you are addressing the immune system. And if you go to vaccinology conferences, I would say 70% of the presentations are actually related in some form or other to particles, following the paradigm here for novel vaccines where you combine the vaccine, be it a recombinant protein, be it a vaccine DNA, with some danger signals, so some ligands, some adjuvants, ligands for the toll-like receptors that Andy Clark was referring to before. So, and you combine this in some particulate form that is recognized by the immune system to be foreign. Now, these danger signals here, the ligands for the specific innate receptors, like for the toll-like receptors, they are indeed, to my opinion, a far cry from the dirty little secrets of Janeway, like he described adjuvants at the time. Talking about toll-like, here you have an overview of all the adjuvants, and here you see the toll-like receptors being expressed on the epithelial membrane, on the membrane of the cells, or in the endosomes, and there are other pathogen pattern recognition receptors in the cytosol, like these NOT1-2 receptors. Now, talking about toll-like receptors, I don't know whether you know, but this lady here, Christine Nüsslein-Vollhardt, received the Nobel together with Wieschaus and Lewis in 1995 for embryonic development, but she also contributed to the discovery of the first representative of the toll-like receptors. Now, you may want to ask yourself why this is called toll-like, what's toll has to do with this. Now, some people think that this is the toll you have to pay to enter the highway or something, but that's not the case. Urban legend has it that as Christine Nüsslein-Vollhardt and her group was working, I think, at Göttingen in Germany, so then they first discovered the toll-like receptor, the first one, they said, oh, toll, which means in German, nice, very nice, or great. So this is the urban legend, and I wanted to know whether this is really true, so I went to the source. I sent an email to Professor Nüsslein-Vollhardt almost exactly 2 years ago, and I asked her to verify or confirm the story, and her answer was, stimmt so ungefähr, yes, this urban legend is about right, so now you know. We're working, in my lab, we're working on having these nano vectors in vaccine formulations using these particles together with a DNA vaccine against tuberculosis. We use the AG85A protein expressing DNA vaccine together with two pathogen pattern recognition receptor ligands, the TLR9, which are actually the CpG sequences of the pDNA we include in the particles, and a NOT2 ligand, the MDP, which then goes intracellularly and activates this pathway there. So we have basically two independent pathways that are activating or should be activating the immune system as adjuvants, very specific adjuvants. Now, as I said, we're using these DNA vaccines here expressing this AG85A from Mycobacterium tuberculosis. And we're using different delivery vectors. One is a nanoparticle formulation based on some biopolymer here that we modified to some extent, together with the vaccine formulation lab of the WHO in Lausanne. We are using the squalene in water nanoemulsion together with DOTAP and a cationome nanoemulsion. This is commercially available as artificial tears for the treatment of dry eye, so we use this as well. And here you see some electron microscopy images of the formulations. Now, we assess the immune responses in a couple of ways. So first of all, we used in vitro methods in order to look at antigen-presenting cells. We looked at the macrophage cell line and the response we're getting from this. In vivo, we looked at T-cell response after restimulation of splenocytes there of immunized mice. And we used, and we looked at total IgG response after vaccination. Now, first of all, the in vitro experiments, here we used the nanocarriers together with the DNA and the MDP. Toxicity was okay, was tested in a macrophage cell line, so there was very low or absent toxicity shown for all formulations. We had then the immunogenicity in terms of TNF-alpha release, and here we saw that all the systems we were using actually gave some significant increase in TNF-alpha release from these macrophages, and indeed this was an additive effect of the DNA, so the TLR9 receptor ligand plus the NOT2 receptor ligand that was not synergistic but additive in effect. We did also show what the contribution of the NOT2 pathway was, so we blocked the NOT2 pathway by giving gefitinib, and you see here that between 38 and 20% contribution of the NOT2 pathway contributed to the release of TNF-alpha we saw from the macrophages there. Now, then we went in vivo. Here we used, until now, there we have the results. There's another study going on. Nanocarrier with a pDNA alone. To make a long story short, from the Humora response, or the IgG, we saw from that cationorm and TMC, the particles here, gave higher IgG responses than non-vaccinated mice. TMC DNA was then higher than pDNA in solution. We got from the cellular response interferon gamma. We saw the release from T cells by pDNA, cationome DNA, and TMC DNA. Especially this emulsion here, we were very surprised to see that this actually suppresses cellular responses in our experiments, and the cationome was better than other nanocarriers, so this thing that you can buy in the pharmacy and mix it with DNA was actually better in this case than the other nanocarriers. A last slide on this. This is an ELISpot data for the TMC nanoparticles, which actually follows the same results we saw in the in vitro study. So we have an additive effect of the TLR9 receptor ligand plus the NOT2 receptor ligand, and this follows the same mounting evidence and mounting response as we saw in the in vitro studies. One more thing before I let you go. As we have seen TLR, toll-like receptors, the innate immune receptors are actually involved in inflammatory processes. Now this is the lung, the airway epithelium from a normal biopsy. And this is from an asthmatic biopsy. So you see here these tight junction complexes between the epithelial cells are nicely expressed, whereas they are disrupted in the inflammatory stage. Now, we are developing our vaccine delivery systems, which are decorated with toll-like receptor ligands, to be aerosolized and inhaled into the lung, to have like a mucosal vaccination by the pulmonary route. So we were asking ourselves, what are we doing if we expose the bronchial epithelium, the pulmonary epithelium, to toll-like receptor ligands? And is there maybe a connection between the expression of these innate immune receptors and the permeability through this epithelial tight junctional complex? When we incubated a human bronchial epithelial cell line with toll-like receptor 2 ligands, we saw a decrease in permeability of paracellular markers. We saw an increase in transepithelial electrical resistance. So obviously there is some interaction between the toll-like receptor ligands and this tight junctional complex. So we got into this, and indeed when we incubated with TLI2 ligands, 2 actually we used, we saw an increase, a significant increase of the mRNA here of Claudin 1 and ZO1. Increase meaning for Claudin 1 it went through the roof basically, and you also see this on the confocal pictures here that are shown here. Also happening on the protein level, we looked deeper into the process that is leading to this, and we saw that this is because of the activity of an atypical protein kinase Z-zeta that is involved in the process that is recruiting in the end Claudin 1 and ZO1 to the tight junctional complex, increasing decreasing permeability and increasing the tight junctional complex. We also did this in an inflammatory model in vitro again. We also did this in primary human epithelial cells, and we see the same thing for this concept. Now, summing up, I hope I showed you for Biopharmaceutics 2.1 as far as these complicated nanomedicines are concerned. You should not only look at this, but the immune system plays a very great role in whatever happens to these nanoparticles, whatever happens for the pharmacokinetics and biopharmaceutics of these nanomedicines. I'd like to acknowledge everybody who worked with us and supported us from a financial point of view, and I would like to close with a quote by George Bernard Shaw from The Doctor's Dilemma. Thank you very much.